I will be talking today about the role of microglia. But before I get started, I just would like to remind everybody that we have our FBI neuroscience retreat tomorrow and would encourage everybody to zoom in. We have a lot of really exciting talks from students, from postdocs. Eric and Paul and George Huntley will also open the meeting, and also please have a look at all the student blitz talks and vote for the best student and postdoc talk. They did an amazing job, and I'm kind of really excited to see that all. So now I will start to, let me go to my next slide. All right. So I will be talking today about neuron-microglia interaction and its role in brain function. It's something my lab is working on now since several years, since I started in Mount Sinai. And if you think about brain in general, brain function, we frequently think a lot about neurons and the circuitries that contribute to behavior outputs and other things. But half of the brain cells are non-neuronal cells that are there to support the neuron as one of a very exclusive client cell, and basically provide the neuron with all of the needed nutrients, buffer, support, and other means so the neuron can really execute its function. And I indicate kind of the key glia cells here, which are the oligodendrocytes, whose main function is to help neurons to really time the electric activity outputs properly, as well as keep long axons alive by feeding them. The astrocytes, which are part of the tripart synapse, regulate neuronal activity, and by themselves have the ability to communicate by gap junction and calcium fluxes. What I will be talking today exclusively is about microglia and how microglia communicate and regulate neuronal function. And so microglia is a special cell. It's not only because it's small, it comes from a different lineage. So the microglia are invading the brain early during development. They come from the yolk sac, from a myeloid progenitor cell. And this is a beautiful image that Anna Badiman, my student, did when she was rotating in the lab, where she shows the dissemination of the green myeloid progenitor cells throughout an embryonic body. And you can see that some of those cells start to invade the brain parenchyma. And this happens really early, around the time when neurogenesis is starting, and the blood-brain barrier will close, and these cells will become the progenitor of the later microglia cells. And they will stay in the brain as a population without any supply from the periphery, and will be a self-renewing population that is sitting in the brain. And these are the brain tissue-specific macrophages or microglia cells. So since they kind of develop really tightly together with neurons, is shown here at the timeline in brain development. This happens before astrocytes and oligodendrocytes are even formed. So we kind of think or postulate that they may have a special connection, let's say, to neurons. And then when we look at the numbers in an adult brain, the distribution, I mentioned neurons make around 50%. It can be different by species. So they're slightly different kind of distributions, but overall, these numbers are rather conserved. You have 20% oligodendrocytes and astrocytes, and only 10% make microglia on average. This already tells you something important that I would like you to keep in mind. It's not that every neuron has a microglia. There's at least a 5-to-1 ratio difference, and that may, I would just like you to keep that in mind. So microglia, what they do, so kind of, they were long thought to be just sitting in the brain, waiting for damage or infection to respond to that and then protect the brain. And one of their main function is the protection against pathogens. However, in 2005, two groups kind of changed really the way how we think about microglia by for the first time showing how microglia behave in a live brain doing live imaging. And what became obvious and really captivated many of the scientists is that microglia are by no means quiescent. And this is shown when I start the video here. You see the baseline microglia activity in the brain, and it's one of the most active cell types in a way in terms of moving the protrusion of the cell back and forth. And you can appreciate all this movement that happened in the microglia. Now, this is kind of the normal state, as we call it. However, the moment damage happened to the brain, and this is a laser injury now that has been placed in the brain tissue, you can see that microglia responds rapidly injury and completely change their behavior. They become proliferative, they migrate towards the site of injury, and they build something which we can almost see as a barrier around the injured region. And that's shown here. So this binary function of microglia, kind of quiescence, but we call it surveillance now, versus activated in response to injury, is one of the major functions of a macrophage-like cell in protection of tissue homeostasis. On the other hand, however, many groups in the last 10 years have shown that there's more to that. It's not just the protection, but there is a function of microglia that is more intimate in terms of modulating neuronal activity, and neurons have a strong influence in modulating microglia reciprocally. So this is something which is really kind of in the focus of what's the role of microglia modulation and neuron communication for normal brain function. Moreover, since kind of even before we discovered microglia as microglia, Alois Alzheimer and Nissel in their stainings found that patients with neurodegenerative disease show an activation of an unusual cell. They called it a glue cell or kind of a different type of gitter cell. This turns out retroactively were the microglia that are activated in many of the neurodegenerative diseases as well as during aging, and that change their morphology and their activity in those diseases. And this is shown here, where you see a wild type cortex, and underneath in green are the microglia, and underneath you see an Alzheimer diseased mouse brain with the plaques, Alzheimer plaques in red, and the microglia become highly activated and change their morphology completely. So this kind of feature that microglia become or change during aging, during neurodegenerative disease, has been widely accepted. And there were several really nice publications that describe kind of the functional and transcriptional state of this neurodegenerative or disease-associated microglia shown here. This, until then, was thought to be more like a consequence of neurodegeneration. However, as Alison had a beautiful talk 2 weeks ago, where she discussed that many actually of the myeloid genes that show mutation or SNPs are frequently, or some of them are exclusively expressed in myeloid cells, suggesting there might be more than just a response of microglia neurodegeneration, but they may have actually a causal contribution to the disease, particularly to late onset Alzheimer. And moreover, there are studies from other diseases where somatic mutations in microglia or myeloid cells, for example, the BRAF, the constitutively BRAF signaling mutation that has been done by Frederick Geisman, this mutation alone is sufficient to drive microglia-mediated neurodegeneration. So the idea that microglia can be causally involved in contributing to neurodegenerative diseases is something that really changed the field in the last 10 years. So thinking about this very tight connection between neurons and microglia, we know that neurons are one of the most diverse cell types in the body. They're diverse in morphology, as it has been, we know, since Cajal and after. They are very diverse in their function, electrophysiological function, which became clear with the discovery of the patch clamp and recording techniques. And then within the last 10, 15 years, we understand much more about their molecular uniqueness. And this is due to several techniques that have been developed, including this ribosome profiling technique that I was very fortunate to work on together with Miriam Hyman in Paul Gwingert and Ned Heinsleb during my postdoc, that allowed us for the first time really to get molecular identity or transcription patterns that are unique or enriched in the specific cell types we are interested in. And this is just shown as an overview here, where the red brackets are genes, group of genes that are enriched uniquely, for example, in the Purkinje cells, versus all the other genes are more or less shared between the cells. And that allowed us to see at the transcriptional level what are the genes that may convey the unique function to specific neuronal cell types. And many of these genes are ion channels or unique transcription factors. But in addition to neuronal functional genes, we also started to see genes that are involved in communicating with glia cells. So there's region-specific and cell type-specific differences in molecules that are important to communicate with microglia, for example. And so that brought us, when I started my lab, that kind of put us a potential hypothesis forward if microglia actually match or adjust to the neighboring neurons. And there were evidence in the literature since many years that microglia in different regions have very different morphology and density patterns, that's shown here on the left side. And so we postulated if it's possible that microglia neurons are actually going into kind of a more intimate interaction, and microglia will adapt to the needs or the specific features of the neurons that are close to them. So to set out to test that in the lab, we started a project that was spearheaded by Pinar Ayata, who joined me after she did her PhD in Ned Heinz's lab, and we chose to look at microglia in different brain regions. So the first kind of the choices we made is we chose 2 brain regions where neurons generally were more homogeneous than, let's say, in the cortex. So we chose the cerebellum and the striatum. And in the cerebellum, let me just see. In the cerebellum, it's 90% of the cells are granule cells, versus in the striatum, 90% of the cells are medium spiny neurons. So in a kind of heroic attempt that looks really easy here, we adapted the trap technology in order to express the ribosome GFP label specifically in microglia. There were a lot of failures. We never managed to make this genset together kind of a direct microglia line. So we are using now a Cre-dependent approach to induce L10A expression in microglia. And this allows us to label microglia specifically. And we can follow that by looking at ribosome associated profiles. This is a big advantage if we study microglia, because we cannot really isolate the cells very well without activating them. So this helps us to bypass any of this dissection and isolation induced activity of microglia. And so when we look at the 2 different brain regions, what we found is that while this microglia share a large number of genes that are microglia kind of or microglia, pan-microglia genes, how we call it, there is a significant fraction of genes that are enriched in one versus the other brain region. So while this striatum shows many genes that are involved in actin polymerization, GTPase signaling, and other signal transduction functions, and we call them the surveillance genes, the cerebellum microglia were really enriched in genes involved in phagocytosis, endocytosis, and very different functional processes. So first, Pinar had to make sure this is not just driven by 1 or 2 cells in those regions. So what she did is she used the nuclei that are labeled in the ribotrap mouse because of the GFP, because ribosomes assemble in the nucleolus. So we can also sort the nuclei of the same mice. And she looked if this is a common pattern of striatum versus cerebellum microglia. And this is shown here with single cell analysis. Each line is a single cell. This is still using the Fluidigm deep sequencing approach. And so what she sees is that the striatal microglia share the specific pattern among each other, versus cerebellum microglia share this different clearance gene expression pattern overall. So that means that in the brain, microglia in different regions show differential transcription pattern, and it raised the question, is this associated with functional specification as well? So what Pinar did first in collaboration with Anna Pimenova and also Oleg Butovsky, she isolated or in vivo injected, kind of exposed this microglia to dying cells. And she looked if microglia in the cerebellum, since they express this pattern that is suggestive of phagocytosis and clearance, have an increased capacity to clear dying cells in vitro or in vivo. And in both cases, it showed a 3 to 4-fold increase of the capacity of cerebellum microglia to remove dying cells over striatum microglia. So that suggested that the cells are not only transcriptionally different, but there's a functional specification in a brain region specific fashion. So Pinar wanted to know, what are these cells eating? And she saw in the cerebellum that the phagolysosomes here indicated by CD68 staining are actually filled with DAPI material, suggesting that cerebellum microglia are eating whole cells in the cerebellum. However, when she looked for dying cells in the cerebellum, she could never see dead cells, neither by caspase nor by tunnel staining. So we remember that this is an actual feature in any tissue where macrophages are present, that these cells clean or remove dying cells frequently long before they can be detected. And so when Pinar then removed from those brains all the microglia, she suddenly saw an accumulation of dying cells only in the cerebellum. And so to make sure that these cells in the cerebellum are not dying because she's removing microglia, she also rendered the cerebellum microglia inactive to phagocytose by knocking out AXL MERTK phagocytic receptors, and she saw the same phenomenon, which suggests that in the cerebellum, unknown to us kind of earlier, there's a slow grade ongoing cell death that starts early early in mouse adulthood and is continuing throughout life, which was really exciting for us because it was not something that we were really aware of. The cerebellum is not a region of neurogenesis as we have in the hippocampus or the olfactory bulb, where we can observe the same phenomenon, that dead cells start to accumulate if we remove microglia or render them inactive to phagocytose. But the cerebellum is not really neurogenesis, so it indicates that cerebellum neurons start to die basically shortly after in adolescence already at a low grade. So that is suggesting that the phenotype of the microglia in the cerebellum is really driven by the presence of dead cells. And that looking at microglia can give us actually a lot of information about the cells or the neuronal environment that they are sitting or placed in. To be really sure that this is an adaptive behavior and it's the dying cells, the dying neurons that instruct the microglia in their clearance activity, Pinar did a nice additional experiment which allows us, so using a pharmacological agent called CSF1 survival receptor inhibitor, or PLX, that's the company who makes it, we can in an adult mouse deplete all microglia within a few days. If we stop giving the drug in the food, this microglia will rapidly reproliferate and come back to the region. And within 30 days after the initial ablation, morphologically, we can't differentiate if this mouse has ever been depleted of microglia because they look basically in density and morphology exactly the same way before the ablation. And Pinar followed these cells at the transcriptional level, and what she found is that the separation between the two cell types is clear. So after the deletion at 5-day recovery, these cells are very different. They express a lot of proliferation and activation markers. However, by day 30, the transcription profile is almost back to what they were before the ablation. So we do believe that even if there's a small fraction of kind of targeted subpopulation in each region, but a lot of the transcription and functional specification is driven by the environment these microglia find themselves in. So going forward then, it raised the question, what are the mechanisms that drive this neuron-specific microglia specification in other brain regions, such as the striatum? For the cerebellum, we think it's the neuronal death. So for the striatum, we were thinking about what could microglia be sensing? And one of the things we always wanted to check is if microglia actually sense normal or baseline neuronal activity and do respond or sense to changes in those activities. And what Pinar did, an experiment, is, so we were thinking a lot, how can we change neuronal activity without perturbing microglia? We can't really use viral injections, we can't really use drugs because many of the neurotransmitter receptors are also expressed on microglia. We don't want to perturb the brain in any way. So we use a genetic approach of the DRET activation or inhibition by using CNO administration IP. So this way we have genetic mouse models where the designer activated drugs either with a G protein that's activating or inhibiting is expressing CAM kinase to alpha neurons, and we can inject CNO IP and rapidly activate or inhibit the CAM kinase to alpha neurons. And then we look at what happened to the microglia in the striatum. So when Pinar does this experiment, and she does it with a relative low amount of CNO, so these mice are just showing increase or decrease in neuronal activity at a physiological level. 24 hours after, when we look, we see already indications that microglia are changing morphologically, and in response to increase or decrease activity, that the responses are differential. So following that observation, Pinar then looked 2 hours after the induction or the inhibition of neuronal activity, how do striatal microglia respond? And what she saw at the transcription level doing ribosome profiling of microglia in the striatum is that they have a differential transcription response, and that in response to increased neuronal activity, microglia induce genes involved in chemotaxis, actin polymerization, and signaling. versus in response to inhibition, it's very different signaling pathway that would suggest potential phagocytosis or NF-kappa B activation and cytokine release. So these data strongly support the idea that microglia sense and respond to changes in neuronal activity. So they have an understanding of a normal physiological response. And that raised the question, do they also reciprocally respond to these changes and are able to modulate neuronal activity in retrospect or retroactively? So when we think about neuronal activity or circuitry activity, we frequently think mainly about neurons. So where would we place the microglia in the circuit and what could the microglia be doing? So to first of all address if they are involved in anything related to neuronal activity modulation, the simplest approach is just to remove the microglia and see what happened to a given circuitry activation, what happened to a given activity, a neural activity experiment. So a student, Anna Badimon in the lab, what she did, she took this approach on and she ablated microglia again brain-wide using the inhibitor. So the beauty of the inhibitor is that you can do this very short term in adult mice, so there's no issue with neuronal development or any other developmental potential changes. It can be induced within 3 days. By 3 weeks, we see maximal microglia ablation. And when she looks at that, she is basically depleting up to 99% of microglia in the different brain regions. And these mice, however, are normal. And this is something which took the microglia field overall by surprise. This has been first published by Kim Green, that you can lose microglia opposite to losing neurons, oligodendrocytes and astrocytes, and at baseline, your mouse is relatively normal. However, when Anna then started to stimulate the circuitries and induced or kind of used neurostimulants like kainic acid, which will bind, will basically activate glutamatergic activity, picrotoxin, which will inhibit GABAergic tone, or a very specific D1 agonist, which will activate neurons that express the D1 receptor in the cortex and the striatum. She saw that in the absence of microglia, these mice display an exacerbated neuronal response, and that leads in all of these cases to a seizure response to subthreshold level of the drug. In addition to the seizure response, these mice also show increased locomotor activity or other phenotypes associated with the drug treatment. So neurons in the absence of microglia respond much stronger to the different stimulus that are given. So in order to address, because the ablation of microglia all over the brain is not really allowing us to ask how is that mechanistically regulated. So we approach it by trying to develop systems that allow region-specific microglia ablation and addressing which microglia regulate, or how do microglia regulate neurons in a more kind of localized fashion. And one of the tools, or one of the ideas we got that helped us to achieve this, this experimentally, is that the CSF1 receptor, the survival receptor, can get activated by 2 different ligands. And the ligands are IL-34 and CSF1. And different cells in the brain are producing this ligand. So IL-34 is predominantly produced by forebrain neurons, so specifically neuronal produced ligand in the forebrain, whereas CSF1 is produced by astrocyte oligodendrocytes throughout the brain. And this is beautiful work done by many people in the field, including Richard Stanley, and we had a collaboration with me and Marat as well. So this is very established. So in the striatum, when we look by single cell sequencing, this has been done by Andrew Chan in collaboration with Bobby Sabra, we can nicely confirm that neurons produce IL-34 versus the oligodendrocytes and astrocytes produce CSF1, the ligand. So that raised the question, is that kind of, are these ligands region specifically expressed in the striatum? And when Anna looks by in situ hybridization, what she sees that within the striatum, you have this difference between white and gray matter. This is really nicely organized. She sees CSF1 is exclusively expressed by oligodendrocytes in kind of the white matter area, whereas IL-34 is produced by neurons in the gray matter. And this is just the distribution. So this selective expression allowed her now to use a conditional strategy to ablate IL-34 or CSF1 in forebrain neuron astrocytes and oligodendrocytes. And what she found is that ablation of IL-34 leads to specific gray matter loss of microglia, So here's the microglia and the white and gray matter. If you lose IL-34, you lose just the gray matter microglia, and that's quantified here, versus white matter stays normal. And the opposite is true if she knocks out CSF1, where she just loses microglia in the white matter, and this is quantified here. So this allowed her now to ask the question, microglia and the striatum, which of them are modulating D1 neuron activity, and is there a difference? So when she does this experiment, she sees it's really the microglia that are associated to neurons in the gray matter that modulate or suppress the D1 excessive activation, versus loss of white matter microglia had no impact on the response of a D1 agonist and basically looked like a wild type. So the next question going forward was to see if this is even more specific. So Anna wanted to ablate IL-34 in D1 and D2 neurons. There was a chance, although a slim one, that microglia might be that specific, that you have D1 and D2 specifically attending microglia. It does not look like that. So if we knock out IL-34 in D1 or D2 neurons, in both cases we lose around 60 or 50% of the microglia in the striatum. The beauty is this is a striatum specific ablation now. However, when we treat with a D1 agonist, both cases, even if we lose it in the D1 or D2 neuron, are hyper-responding to a D1 agonist, which is suggestive that in the striatum, there's no difference between microglia attending D1 or D2 neurons. However, loss of 50% of the microglia in the striatum is sufficient to have these neurons hyper-responding to a D1 agonist. And importantly, this is highly localized, so we can treat the mice that lack microglia only in the striatum with kainic acid, and these mice will show a wild type-like response. So they really just over-respond to a D1 agonist, suggesting this microglia-neuron interaction and modulation is extremely localized within the brain region. So that raised the question now, how do microglia sense and control neural activity in the striatum? And these are two projects by two fantastic PhD students. They are conceptually slightly different. So Haley is asking if there is a very unique interaction between neuron and microglia and the striatum. And I will talk a little bit about it. The idea is that microglia in the striatum may respond to dopamine in a very specific fashion, and that will allow dopamine-induced cross-communication between microglia and striatal neurons. Versus Anna is looking at a more generic response where neuronal activity leads to the release of ATP, and that may inform the microglia about the activity state and then reciprocally lead to modulation of neuronal activity. So very briefly, for Haley's project, so what we do see, and that comes from Pinar's single cell analysis, that there is a population of microglia in the striatum that express a D1 receptor. And this is striatum specific. We don't see this population, for example, in the cerebellum. And moreover, the population of the D1 expressing microglia that's indicated here is kind of express a set of functional genes that suggest that the D1 expressing microglia might be even a specific subpopulation that may have modulatory activity in terms of neuronal communication. So Haley nicely confirmed using all possible methods from in situ hybridization to protein staining that microglia in the striatum indeed express, and lineage tracing express a D1 receptor. So encouraged by that, she went ahead to ask the question, what's the role of a D1 receptor expression in microglia? And to do that, she generated mice that lack D1 receptor specifically in microglia in an inducible fashion. So she let the mice develop normally, and then she can take out the D1 receptor on microglia in adult mice. And this is shown here by Western blot, which gives really the nicest confirmation. So she sees in the control striatum, but not in the cerebellum, that isolated microglia express D1 receptor that is specifically depleted if she deletes D1 receptor in a microglia specific fashion. And to support the functional role of a D1 receptor in microglia, she then challenged the system with dopamine, with D1 agonist or cocaine. And what she finds is that the loss of the D1 receptor on a subpopulation of microglia is sufficient to modulate neuronal responses to dopamine. And so what Haley is looking at now is, how is that possible? What are the microglia actually doing in response to sensing dopamine by themselves? How do they modulate the neurons? and what could be molecular features that actually instruct the microglia to express the D1 receptor in the first place, which is a really exciting question. This is work in progress. So, and Anna, on the other hand, looks this is a more complete study. Anna is looking how can microglia sense neuronal activity if they respond to the release of ATP, for example. And it's known that neuronal activity, cortical striatal projections, activation leads to release of ATP from the neuronal synapse together with the neurotransmitter release, as well as astrocytes that are participating in the synapse can release ATP in response to neuronal activity. So the neuronal activity induced ATP release at the synapse has been well established. Microglia, on the other hand, express receptors that can sense ATP. And one of the receptors is called P2RY12 that is expressed on microglia exclusively. And that has a affinity to ATP that is not activated at baseline, because baseline levels of ATP extracellular in the striatum are rather low. They're around 3 to 5 nanomolar. But then in response to neuronal activity, they can go into the micromolar locally at the synapse, and the receptor P2RY12 on the microglia can recognize that. So to see how microglia respond to neuronal activity and ATP release, Anna collaborated with Hiro Wake in Japan, and we look at in vivo imaging, activation of neurons, what does it to a microglia morphologically. And so what we find by in vivo imaging, that increased neuronal activity leads to a outgrowth or a protrusion of a microglia towards the synapse, and this is quantified here. And this targeted outgrowth of a microglia protrusion into or towards the synapse is dependent on this ATP receptor called P2RY12. And when Anna is inhibiting that, what she sees it abolishes the targeted movement of the microglia towards the synapse. And this P2RY12 dependent recruitment of a microglia to the synapse is functionally important, because if Anna inhibits P2RY12 pharmacologically or genetically, what she sees is that these neurons display the same hyperresponsiveness as if you just would not have the microglia at all. So this is very indicative that the microglia not only responds to ATP, but that this behavior or this localized recruitment is modulating the neuronal circuitry. So ATP, once it's released, is rapidly broken down into AMP and adenosine. And adenosine is one of the major neurosuppressive modulators in the brain. The enzymes that break down ATP to adenosine are ATP hydrolases or ectonucleases, and CD39, or called ENTBD1, is highly enriched in microglia and is expressed highly on microglia in the striatum. And that's just shown here, the microglia population by single cell sequencing. So the question is, can microglia physically actually convert ATP and adenosine? And to address this question, Haley and Anna did first an in vitro experiment where they isolate microglia, add ATP to a microglia culture, and then within an hour measure by HPLC the generation of adenosine. And microglia nicely break down ATP to ADP, AMP, and adenosine. And this synthesis of adenosine requires CD39 and CD73 activity. If they inhibit either CD39 or CD73, we lose the production of adenosine by microglia. So this data suggests that microglia can process ATP to adenosine, and moreover, in collaboration with Erin Calipari, Anna showed that if she depletes microglia from the striatum, you see a significant decrease in extracellular adenosine levels in the striatum at baseline. So what does adenosine do, or is that important, the CD39? So to prove that the synthesis or the conversion of ATP to AMP and then adenosine is truly mediated by microglia, Anna did a genetic experiment where she removes CD39, the key enzyme in the process, the rate-limiting enzyme, specifically in adult microglia, and she sees that leads to a decrease in adenosine and increase in seizures in response to D1 agonist, suggesting this conversion by microglia is the key step in this regulation. Furthermore, once adenosine is produced, and this is work by many labs, It can act in the striatum pre and postsynaptically on the A1 receptor, which is the receptor with the highest affinity for adenosine in the brain. And the idea is that it inhibits synaptic transmission by A1 receptor activation and suppression of glutamate and vesicle release in the presynaptic side, and by inhibiting the response, the signaling response in the postsynaptic D1 neuron, where it counteracts PK activity. So to show that the A1 receptor is important, Anna did kind of a pharmacological and genetic approach. So first she pharmacologically inhibited the A1 receptor at baseline, which has no effect, and then in combination with the D1 agonist, which reproduces the increased D1 neuron activity nicely. And then she even went further to knock out the A1 receptor specifically on the postsynaptic D1 neuron, and that was sufficient to cause the response she has seen. So the idea is that microglia produce adenosine and then suppress the D1 response for the propagation or kind of the synaptic transmission and the response in the D1 neuron per se. So that implies, if this is all correct, that implies that we should be able to rescue it by giving microglia ablation or the inability of microglia to sense or generate adenosine by just activating the A1 receptor downstream of the event. And so Anna did that here genetically. So she was able to show if she ablates microglia in the striatum, which leads to this increased response to a D1 agonist, this is rescued if she combines this with an A1 agonist. If microglia lacks CD39 or P2R12, pharmacologically, she can also rescue this with an A1 agonist combination, and the mice show kind of a wild type-like response to the D1 agonist. And moreover, the knockout of CD39 is equally rescued by giving an A1 agonist together with a D1 agonist. So collectively, we think that microglia are recruited to active synapses very specifically and localized to the activated synapse, and then lead to a local synaptic specific production of adenosine that will lead to suppressing of kind of the propagation of synaptic transmission and activation of D1 neurons downstream, and that this feedback, negative feedback mechanism is essential to kind of to allow normal signal response in the striatum. And so that kind of in summary, what kind of the first part or the 2/3 of my talk so far basically want to convey is that we do have a brain region specific microglia-neuron interaction that's important for region specific functions. We showed that in the clearance fact in the cerebellum, but in the striatum, the main function is neuromodulation of striatal neurons. And so the question is this important? Are these unique features of those cells, are they really important, and are there perturbations that will involve these particular pathways? So what Anna, or kind of what this kind of the bigger question for the lab is, does aging or diseases override this microglia-neuron specification? So, and what are the signals that will allow a microglia to specify or to change their activation pattern in response to which cues that are released by the neuron? So this is kind of showing the lab program a little bit, where Anna, Haley, and Philip are trying to understand what are the signals and the normal mechanisms, how microglia and neuron interact and modulate each other, versus that can change if in the course of aging or infections, where we know a lot of the signals that are easily perceived and can activate microglia are produced, like type 1 interferon, cytokines, senescence-associated secretory pathways. And Andrew is looking at that in a specific fashion, where he asked the question, if we have peripheral viral infection, like from the flu or the virus we are dealing with now, he sees that microglia are responding to peripheral signals, and they're responding in a brain region specific fashion. And that may actually allow or modulate some of the behavioral responses we are seeing in response to peripheral viral infection. Versus Pinar, Sarah, and Sahil, and the former postdoc Melanie, were more interested what are the kind of the new ligands or the new signals that happen during neurodegeneration, where microglia are suddenly exposed to toxic proteins or phospholipids, plaque, artificial kind of material, and microglia has not been exposed, an increased number of neuronal death. How will these signals override or affect this microglia-neuron specific interaction that I just tried to present to you, that this is really important? So one of the hints that we get that these specifications are important, play a role, not just in the healthy brain, but also in disease, comes from Anna's work, where she sees that microglia that become activated due to several different stimuli that can range from inflammatory stimuli to all or many of the neurodegenerative diseases. A microglia, when it becomes into kind of a reactive microglia, activated microglia, is characterized by downregulating exactly those key signaling pathway and receptors that Anna just kind of showed us before that are important for modulating neuronal activity regulation. So in response to LPS, for example, CD39 and P2R12 are dramatically downregulated in microglia, and the same is true for Alzheimer Huntington mice, where we know microglia activation occurs. So is that playing a role in those diseases, or is there contribution of microglia, loss of microglia-neuron interaction that may lead kind of to changes in neural activity? And we are very excited because both of these models, it can be kind of the LPS-induced neuroinflammation, where it's more from a peripheral inflammatory condition, or the 5xFAD mouse model, which is an Alzheimer mouse model that develops plaques and changes in neuronal survival and activity. In both cases, we see that the downregulation of CD39 and P2R12 is actually associated with an increased responsiveness to a D1 agonist that can be rescued in both cases by giving an A1 agonist to kind of normalize or restore the normal function of a microglia. So it's possible and very likely that changes in microglia activity not only kind of is a gain of function that is neurotoxic, but also just the loss of their normal function, their normal neuromodulatory function contributes to the disease development and some of the behavioral features associated. And both LPS and 5xFAD injected mice are susceptible to seizures and hyperresponsiveness. So Anna would postulate that this is due to in part, the perturbation of the microglia suppressive pathway in these mice. So that leads us to kind of a model where, and that's something I kind of was trying to build throughout the talk, that there is a match, a functional match, a transcriptional match, a match between a neuron and a microglia in a given region. And what I didn't mention, and because it's kind of microglia in the brain usually don't move. So if a microglia settles next to a neuron, it will sit there for most of its life. And even the new cell, the kind of, if it goes into division, the new cell will take a very similar area where it will sit. So this kind of lifelong, almost a cohabitation or matches, we basically would propose that this also leads to a functional specification. And then what happens if this match is perturbed? If neurons during neurodegenerative diseases are changing, how will this affect the microglia response? Or if microglia are getting activated because of inflammation, infection, that has not directly anything to do with the neuron, how will this change in microglia activity affect neuron survival and function? And so to ask this question in kind of the most crystallized or the most non-perturbed way, a postdoc of mine, Melanie von Schimmelmann, who basically designed a study where neurons are changing their identity and their specification just mildly by interfering with their epigenetic regulation. So this leads the knockout of an epigenetic modulator called polycomb repressive complex 2 in striatal neurons leads to a small amount of changes in neuronal gene expression without any other pathogenesis. So there's no neurodegenerative kind of other development like plaque or fibrils or anything. It's a pure gene expression switch in those cells that make a striatum neuron still function and look like a striatal neuron, but it leads to upregulation of genes that are usually not expressed on those cells and just slightly changes the specification pattern. And this is enough to cause those cells where it's purely a de-differentiation transcriptionally of a neuron to induce neurodegeneration and associated activation of microglia. So we don't even need pathogens or plaques or anything. It's sufficient if a neuron intrinsically changes its gene expression profile and slightly its function for the microglia to pick that up and get activated. Conversely, what Pinar showed, the same is true if you do the same trick and change striatal microglia and make them slightly more cerebellum-like or what we do if we knock out polycommon microglia, they start to induce some of the clearance genes. But they are not changing overall their microglia fate. They are just becoming slightly different in their activation or specification pattern. And again, this is enough to have negative effects on neuronal morphology, behavior, and induces the seizure phenotype in this mice. So that brings us to kind of this model that small changes that interfere with kind of the match between the microglia and the neuron may be driving or may be causing neurodegenerative and neuronal dysfunction. And that, if you see conceptually, is very close to a concept that we in the immune system is frequently postulated and studied. It's the self versus non-self recognition. And so that a microglia and a neuron, when they match and recognize that self, that they are in a normal interaction, this can be disrupted, and the microglia may recognize the neuron as a non-self or a mismatch and thereby go into attack, and vice versa. If a microglia changes, this will have negative effects on the neuron. If you drive this thought provocative a little bit further, you can even think about neurodegeneration as a form of an autoimmune disease where the microglia cannot recognize a neuron anymore as self and start to attack it. And so this is a concept we are actively exploring in the lab, which will also kind of opens new ways how we would like to interfere with neurodegeneration. In a way, we will not be able to change the neurons or their, let's say, despecification, but what we can do, we can identify the signaling pathways and transcriptional profiles of the microglia that become in this inflamed or activated state, and find ways to either by targeting the signaling pathway, and that's what Pinar is working on, or their epigenetic switches that allow the suppression of this inflammatory response, with Sarah and Sahil are working on, to restore and bring the microglia back into the kind of self and away from this non-self attack of a neuron. So that's kind of where I will finish. So what I hope I have done in kind of the past 50 minutes, I hope I was able to excite you to think about microglia, to convey to you the concept that microglia and neurons are intimately interacting and regulating each other, and that we should think about them in terms of neurodegenerative diseases in a way how we can kind of restore their normal function, and that may be beneficial to the diseases. And these things may all happen on top of it in a brain region specific fashion that may explain some of the changes we see and why certain diseases have maybe kind of why we can see some region specificity in some of those diseases could involve activation of microglia as well. So I will stop here. This is kind of the most important slide. I would first of all like to thank my lab, and it's particular, I think, in times like now where you really see the passion and the enthusiasm and kind of also the togetherness of a lab, and I'm super proud of all of them, how they dealt with the last 5-6 weeks, and how there is really no decline in their motivation and excitement about science. I would like to particularly like to thank Anna, Pinar, and Haley for their kind of work on microglia activity modulation, and Sarah, Andrew, Sahil, Philip, Diogo, and Zach, who all contribute greatly kind of to all of the project which I presented. Former members of the lab, I would also like kind of to use this moment to congratulate Anna and Pinar. Anna will start her postdoc at Rockefeller in the summer, and Pinar will start her own lab at CUNY, and I'm super excited and proud of both of them. I would like to thank my amazing collaborators at Mount Sinai. We have a very intense and fruitful collaboration with Alison. I would like to thank Eric and Paul, as well as Bobby and Miriam, with whom we are working, and Adolfo on the flu project. My late mentor, Paul Gwingard, who really kind of imprinted many of his features and preferences and his sense of humor in me, and I will be internally grateful for everything what I learned from him. I catch myself frequently kind of displaying certain features that I always observed on him, which is a lot, kind of, it's very touching for me and very funny. I would like to also thank collaborators and Harvard, Oleg Butovsky and Francisco Quintana for their work on the microglia, Marco Colonna, Hiroki Wake, and Eo, Viviana, who did a lot of the work. I have not been able to show it, but in this Anna's project, Joy Schaefer, Aaron, and Jim Surmeyer. And I would like to thank my funding. This kind of novel focus on microglia was really possible because of the director award that I got when I started my lab. That made a huge difference. And I would been kind of particularly honored for the NINDS Mentor Award, which meant a lot to me, which was kind of suggested by my mentees. So I have time now for questions, and it would be really nice kind of to, you know, to get some questions. So please don't be shy, and let me know what you think.
One question, and I don't want to go into semantics, but when you talk about the mismatch,
so before you go, just before, can I just say what a wonderful job you did, and that was a great seminar. Lovely to see the data. Don't mean to interrupt, but just in case we don't get time to say that, really nice job.
Thank you, Paul.
Sorry, Dana.
Yeah, super good job. As you mentioned also at the very beginning, microglia is cell type is highly plastic and therefore highly adaptive to changes in the client cell. So what do you think is actually causing this sort of unexpected mismatch that the microglia cell would not be able to adapt adaptively, and
so I didn't make that clear enough. So microglia are very responsive to changes, right? I mean, that's the feature. They are the cells that are supposed to respond immediately to any changes in your brain homeostasis. And these are the cells that are supposed to eat away pathogens, eat away debris, and have a typical macrophage function. So for us, the question is, it's almost like a hierarchy of signals. What are the signals that the microglia is getting that will override the homeostatic signals that they get from a neuron? So in a way, we think about it that while in a normal, let's say, striatum, the microglia is deeply engaged with modulating neuron and sensing the activity, the moment you have a stronger and potentially more dangerous signal, that's what will be kind of switching a microglia in their specification state. So you can think about a hierarchy. So a dying neuron will be a bigger danger or will require potentially more attention of a microglia than homeostatic modulation of activity. So in those cases, the microglia will kind of lose their normal interaction with a neuron at the cost of neglecting kind of neuronal modulation, but therefore maybe attend plaque removal, or however you want to think about that. So if we identify the signals and can block them or interfere with them, we think then we can restore the homeostatic function of the microglia.
And this is our great talk. As Paul said, it's really a gorgeous body of work. I have a quick question for you, and I don't think we've talked about this before. When you manipulate the microglia in cerebellum or striatum in the polycomb complex components, do you by any chance affect D1 receptor expression in the microglia?
It doesn't look like, but it's difficult. I can't say that. Because to be really sure with the D1 expression, we need to do the single nucleus sequencing. So what Pinar has done is all trap-based. With the ribosome profiling, because of the affinity purification, we actually do not look at the D1 receptor because we are never sure this is not kind of a background from the neuron. So to really say this, Pinar or Anna or Haley or whoever continues the project would need to single nuclei sequence or do an in situ hybridization. So I actually can't, we don't know. It's possible. It's possible.
Mm-hmm. And can I ask you about the huge differences, striking differences you see in microglia in different sites? I wonder within sites, you get state transitions in microglia, of course. They have different, profiles of gene expression depending on what's going on, even neuronal activity doing that. But within a site, do you think that there's unique populations of microglia with specialized functions that you can begin to discern with this type of single cell approach?
Yeah, so that's exactly what we are thinking. So we think that there are subpopulations similar in a way to immune system, kind of macrophages that may respond, may have, and that's where I didn't go into detail, but that's a little bit where actually the D1 expressing subpopulation in the striatum is also already indicative of that, where you may have particular subpopulation that are more responsive, let's say, to inflammatory signals, and then will mediate a response that may then secondarily involve other microglia. So we do think we are dealing with subpopulation. The single cell sequencing of microglia, so microglia, and I think Haley and Anna and Pinar pointed it out frequently, we get a very low read overall. Microglia have less RNA than other cells. So you have to sequence really deep in order to allow you by single cell analysis to pull out these differences. But yes, we do think they are there. If you activate or if you give an inflammatory signal, you only see a certain percentage of cells responding first. I think it's just a bit more tricky to even, go further. You need unique markers of the subpopulation that you then can take out the subpopulation or address their function. And that's what we're trying to do.
Fascinating. Thanks, Anne.
I find the hypothesis of neurodegeneration being autoimmune disease very fascinating. So do you think for some of the neurodegenerative disease, they might exhibit similar inflammation features to, like, say, multiple sclerosis?
Yeah.
And are there differences and similarities?
So Behar Ajami has done a really nice job. So in general, so microglia get activated, and this disease-associated microglia pattern, there are a lot of similarities, but it looks like every disease is slightly different in terms of how the exact, let's say, cytokine or phagocytosis profile of gene expression in microglia is looking. So I can't say that they're all kind of, but so in many of the cases, I do think, yes, particularly in MS, I think that's all. I mean, MS is in a way at least an autoimmune disease, and there it's the most fitting in a way. But I do think this inflammatory activation then by signals that may not necessarily be in every case, a neurodegeneration, but might just be sufficient to inflammatory or neurotoxically activate the microglia by inducing interferon pathways, that would be sufficient to attack or to be neurotoxic.
Right.
But it might differ slightly in all of the diseases because the signals that the microglia are getting are slightly different.
So Anne, that was a really beautiful talk and really exciting work. So you had mentioned that you're interested with aging, and so in stage 3 of the hippocampus with aging, in both humans and rodents, there's increased excitation, and it's thought that this is part of the cognitive decline. And I'm curious whether you see an increase in microglia response with aging in this brain region, maybe as a neuroprotective function, or maybe that maybe part of the increased activation of this brain region is reduction in microglia. I'm just wondering if you know what's going on with microglia with that.
I think it's an excellent question. It's exactly a brain region where we do think with age microglia are becoming more inflammatory. We have not looked at it, but this is something we could, we can discuss what would be a good way to look. Intuitively, or it would be suggestive that microglia in that region will downregulate P2R12 and CD39, and I do think there are indications for that, and that could lead to a hyperexcitability of a neuron just because of the lack of microglia-produced adenosine modulation. Yeah, so we do think, but we haven't done the experiments.
Thanks again, Anne. That was fantastic.
Thank you, everybody, for listening.
Thank you, Anne.
And I hope to see you all soon, of course. So
yeah, hopefully soon. Hopefully everyone soon.